In the previous lecture, we did look at the topic of toxicokinetics as what the body does with xenobiotics that get into it. The processes of absorption, distribution, metabolism, and excretion were major steps, major processes that biological systems carry out on any xenobiotic that enters into it. Today, we're looking at the toxic effects, how the substances that get into the body, how they carry out, their toxic effects. I mean, in other words, what these substances do to the body. If you had looked at what the body does with the substance, now we are looking at what the substance does to the body. So that we'll be looking at some mechanisms of toxic effects of noxious substances. So that is generally what toxicodynamics is about, which itself falls under what's one of the branches of toxicology that we saw right from the introduction of this course, which is mechanistic toxicology, focused on the mechanisms of action of toxic substances. So mechanisms of toxic effects, looking at these effects at the molecular level. So these molecular mechanisms simply is referring to the interactions between the toxicant and other chemical entities that are in the body. Even looking at it, say in terms of interactions with biomolecules like proteins, lipids, nucleic acids, so that we specifically say these are biochemical mechanisms. And then, of course, we also look at it at the level of the cell. So we're looking at the toxic effect on cells. Since cells are the basic units of life, any of these effects that we are looking at, we want to look at it in the simplest of life system. Of course, you can even, from this, you can deduce that you can look at it at the level of the tissues. I mean, from the cells, you can look at it from the level of the tissues and go ahead to look at it from the level of the organs before you move on to organ systems and then the whole organism. You can look at it that way. Toxicodynamics specifically, focuses on these three levels: the molecular level, the biochemical level, and the cellular level. So that when you have a substance, a noxious substance, a toxicant, which the biological system is exposed to, that we did look at the fact that you can have some of these substances accumulating in the tissues. So you remember we did look at the concept of volume of distribution, where you have some of the substances having more affinity for the cardiovascular system, staying in the bloodstream more than in the tissues, and some staying in the tissues more. Those ones can result into accumulation in the tissues if they have more affinity for the tissues than the bloodstream. Then, of course, in the tissues, they begin to have biological interactions. They have interactions with biomolecules like proteins and their complexes, like say maybe glycoproteins, lipoproteins, which serve as receptors in cells. That then results into some toxic effect, what is being referred to as perturbation, some alteration of biological processes. So if in a biological system there is a normal function and you have an alteration, that's a perturbation, a disturbance of that system, that can result into some cellular changes to which the cell can react in terms of adaptation. So have some adaptative cellular response to whichever perturbation is occurring with that normal function of the cell. Or that alteration of the normal function of the cell can result into cell injury. So that will be the toxic effect that will occur on that cell. So a group of cells which are in tissues, such injury will become a tissue injury. If those tissues are in some specific organs, obviously it results into some organ injury. Then, of course, systemic injury that can also happen if you are looking at organs that are coming together in a particular organ system. Then, of course, what effect does it have, the toxicant have, generally on the organism? So from the cells, you build up level by level, and then you begin to understand the general effect of both the general and specific effects of these toxic substances. Now, looking at these, effects of the substances on biological systems, we can categorize these into two broad categories of what we refer to as direct acting and indirect acting of toxicants. So what we mean by that, actually we did look at it in our introduction to the course briefly, where we did talk about the fact that you can have, so for instance, this is a xenobiotic. So xenobiotic there, and we can take the first part where the xenobiotic interacts directly with a receptor. So what's a receptor? A receptor you can see the receptor here is shown as a cluster of proteins. Usually it's going to be in the cell membrane. It can be a transmembrane protein, so embedded in the cell membrane, that is going to receive some response, so to say, for cell recognition of this xenobiotic. So the xenobiotic binds onto this receptor. Of course, we did see, I think in general biochemistry, you can have such receptors on the cell surface. You can have receptors also inside of the cell. So that some xenobiotics, which means lipophilic xenobiotics, will be able to penetrate through the cell membrane and then can bind to these receptors. If these are hydrophilic xenobiotics, they will generally be binding to receptors on the surface of the cell, because they cannot easily pass the cell membrane into the intracellular space of the cell. Now, once you have the xenobiotic binding to the receptor, so you now have a xenobiotic, receptor is a non-covalent binding occurring, so that is irreversible. Then the next thing you see is that complex, the xenobiotic receptor complex, may then bind to a biomolecule. So the example which is being shown here is that it's binding to a promoter region of DNA, which means a promoter region of a particular gene which is to be expressed. And once it binds to that promoter region, it will then alter the expression of a particular gene. That is the synthesis of a particular protein. Now, it either inhibits the synthesis of such a protein, or if such xenobiotic receptor complex is as an activator of the synthesis of a particular protein, then it means that there's going to be some vigorous synthesis of such a protein, which perhaps will be resulting into some carcinogenic effect, for instance. It can result into cell death if it is inhibiting the synthesis of an important protein, which is to become some enzyme or a neurotransmitter. For instance, if it is some acetylcholine esterase, which is to you know, participate in movement of muscles. Of course, if you have such xenobiotic receptor blocking the synthesis of acetylcholinesterase, that is going to result into some paralytic effect in the end. But that is the say the direct route. If you have a direct acting toxicant. How about an indirect acting toxicant? So the part number 2, which is down, you see the xenobiotic first gets converted into a metabolite, and it's the metabolite that then causes some toxic effect. So if you look at it step by step, you see this is a xenobiotic upon which an enzyme is acting. So you see xenobiotic enzyme complex, and you have from here conversion of the xenobiotic, more specifically some oxidation reaction where you have the some electrophils, or let's just say a metabolite of the xenobiotic being formed. I mean, that process of forming an active metabolite from a xenobiotic is what is being referred to as bioactivation. We did look at it. I mean, when we were looking at the phase 1, phase 2 reactions, we did see that you have this process of the conversion of a xenobiotic into either an active metabolite or an inactive metabolite. So this is an example of such a thing where you have a xenobiotic that an enzyme is acting upon to produce an active metabolite. Then that active metabolite, as you see, the arrow up is showing that that active metabolite can then bind to a receptor. So from that receptor, metabolite complex, then the reaction path goes in the same manner as the first reaction path for the direct acting toxicants. The only thing is this time around, it is the metabolite that is binding to the receptor, not the xenobiotic itself. Or you have the active metabolite directly binding to a biomolecule such as DNA. So such will be forming what we do refer to as some adducts. It's not necessarily only a DNA. I mean, if it's a DNA, it means that the level of such complex formation is at a genetic level. It can even be at the level of, say, proteins. Let's say it can bind to some enzymes to cause some inhibition of such enzymes. Similarly, if it binds to, as we saw up there, binding to some say promoter region of a gene, either to activate it or either to inhibit the gene from expression, then that will then result into some toxic effects or cell death, just as the direct pathway that we looked at above. So that is direct versus indirect acting toxicants. Now what we'll do is that we'll take some specific examples of these things, looking at, say, direct toxicant effect or indirect toxicant effect, some very specific examples, and look at how these things occur. So the first example we are looking at is well, not so toxic, but just to understand the mechanism, so that we take what we refer to as vanilloid receptors, sometimes being referred to as some transient, receptor proteins, TRP, transient receptor proteins, or sometimes just vanilloid receptors. And these are receptors which are responsible for temperature recognition, heat recognition, like say in the skin cells, for instance, or these receptors are also responsible for the recognition of, the heat you feel when you take in pepper. And I mean, the compound which is responsible for such heat that you feel when you take in pepper is capsaicin. Now, what you see is that in this cell membrane, you have, a cluster of proteins. These 4 proteins, with different colors like pink, violet, green, blue. These are, a complex of proteins which are coming together as the receptor, this transient receptor protein complex, or the valenoid receptor complex, which is embedded in the cell membrane. Of course, it's forming something like a channel through which you can have ions passing through. So it's like an ion channel. You can refer to it as some ion channel. Now, what you see is that these vanillin receptors respond to temperature, heat, so that with temperature, you react that the receptor opens up for ions such as sodium ions, calcium ions, some protons, or hydrogen ions. Or if it is not even heat, if it's not temperature, and but it is the molecule capsaicin, which is responsible for that heat sensation from pepper, that can also activate the opening of such receptor proteins. Now, once there's the movement of these ions into the cell, this results into some changes in potential across the cell membrane, because you have some positive charges moving from one side to the other, and then you have some electrical signal that can pass along, nerve cells, because this is actually occurring in nerve cells. This cell membrane is of a nerve cell, and so there is that signal transduction, electrical signal transduction into the central nervous system, and that signal is then recognized by the brain. So the brain then translates what you feel on the skin as burning, or if it's a sting from maybe some bug or some mosquito, or it's some heat, say from the atmosphere, temperature, or maybe some getting close to some hot substance. You have such a feeling of heat, a burning feeling, or an itching feeling. You do feel that. If it's the tongue, yes, which is tasting pepper, it will be similar sensation that will be translated by the brain. So that's an example of a direct, action of a xenobiotic. Now, we did look at an example of this synthetic estrogen, the diethylstilbestrol. We talked about it as one of these substances that caused some serious problems, where, historically, we did look at some history of some substances, and we talked about the fact that this diethylstilbestrol was prescribed for pregnant women who, even though they themselves, at the time they were taking it, it didn't affect them. Later, it affected their children, particularly, daughters. It affected them in their adolescence or puberty, where they were at high risk of developing breast cancers, and they also had, problems with miscarriages. And what you see in such, mechanism of action over here is that you see, so in the fetal period where the mothers took this medication, you do have some, what is referred to as some epigenetic reprogramming of cellular signals in these germ cells, which were still at their developmental stages at the time that the mothers were taking this medicine. Now, such reprogramming of these somatic cells then resulted into silencing of some specific genes which are responsible for protecting the organism from cancers, and rather activated genes that promote cancer. You see, you have the same substance, silencing some genes, and at the same time facilitating or activating other genes. So the genes which are not the expression of which are not good are rather activated, because those genes, when they're activated, they begin to produce some endogenous substances which then result into some cancers, while the genes that will be producing protective endogenous compounds from cancers rather are silenced. And then this also results into increase in a number of, so let's say, some mammary births where you have breast cancer being initiated. And so then in adulthood there is that high risk of those offsprings of mothers developing breast cancers. And even the sons, we did see the sons were also prone to developing some reproductive abnormalities, disorders. So that is a direct action. You do see it's not a metabolite that is causing this, but it is the drug substance itself which is causing that. Right. Now another example we can look at is we did talk about these two dioxins, particularly when we were looking at, I think we referred to it briefly, looking at some environmental biotransformation. I mean, biotransformation, not in living organisms, but in the environment, where we were looking at some compounds which, upon exposure to, for instance, sunlight, they begin to undergo processes of, say, maybe photooxidation. We did see some of these things, and particularly the production of TCDD from some particular pesticide or from a herbicide. We did look at that. And we mentioned also that this TCDD, I mean, or generally the dioxins are produced from burning, or which a lot of times is from maybe engines, so that you have a lot of dioxins coming from exhaust of fossil fuel engines. You do have it coming from industries that release some gas waste into the atmosphere. You have a lot of dioxins in there. Now you see the picture that is here. It's actually, this is agbogloshi, where you have a lot of these guys indiscriminately burning things over there, looking for all sorts of metal scraps and things in it. And you can see the kind of smoke which is coming out. That is a very, very dangerous, very dangerous activity to get involved into. With all this smoke, it's a lot of toxic compounds in the gas around this whole place. And in fact, the effect of dioxins on the unborn baby is a serious thing. So you see, TCDD as this a dioxin. When a mother gets exposed to such a gas, the smoke coming out, breathes it in, it can actually cross the placental barrier, so that you see that it can result into some adverse reproductive outcomes like inflammation of a preterm inflammation of the unborn baby can result into even infertility in persons, both males and females. And you can have, if for a pregnant woman, this can result into some preterm birth, or even infertility of the child that will be born. That can happen. And you do have, if you zoom into the placenta at the time that such toxic activity is going on, what happens is that you have the dioxins acting as inhibitors to, say, for instance, the phagocytic activity of, say, placental macrophages. These are, some immune cells that fight infections by phagocytosis, so that if these cells are inhibited, then of course the newborn baby is vulnerable to infections. So that of course can be resulting into some miscarriages, inflammations of the newborn baby, and more serious things can happen there. So you can see that you also do have some immune cells like cytokines, interleukins. All of these are inhibited. Some immune factors, they are all inhibited. So you do see that you have TCDD directly inhibiting the immune system. I mean, the growth of the CD4 plus cells. This can result into serious immunosuppression of the not only even the newborn baby, but even for the mother as well. And this is a direct action of the dioxins. We can look at another direct action of cyanides. I don't know if we discuss the example of cyanide being used, historically as a chemical for judicial executions. So, I mean, in the past, I don't know if at the moment there is still any country that practices this kind of thing, where when you have convicts sentenced to death, sometimes they are sentenced to death by chemical execution, and the chemical which is used is cyanide, and cyanide causes what we can refer to as cellular suffocation. And I mean, how does that happen? So you have cyanide. You have cyanide that coming out of some food substances as well. You do know that in Ghana, a lot has been said about cyanide poisoning in cassava. But then it's been known that if you soak the cassava for some time, say maybe overnight or something, you can have those compounds that form cyanides being converted, so that there will not be any problem. Now, I mean, what does cyanide do, or what do cyanides do? So the cyanide goes to inactivate the enzyme cytochrome oxidase. And cytochrome oxidase is involved in a process of oxidative phosphorylation in the mitochondria. I mean, this is an important process of cellular respiration, so that if there is a deactivation of that enzyme, the cytochrome oxidase, what it means is that the aerobic cellular respiration is inhibited, and the condition which is created is an anaerobic condition, where, for instance, you have, say, the formation of lactic acid, the process we refer to as lactic acidosis. I mean, instead of having that aerobic process that we saw, say, in the pathway of glycolysis, where you have acetyl-CoA, I mean, going into the TCA cycle and producing energy, carbon dioxide. Instead of that occurring, then you then rather will be having the conversion of, say, the carbohydrates rather into lactic acid, which is an anaerobic process. So I mean, in the same manner, when cyanide, I mean, I was talking about, say, judicial execution, when say, convicts that are sentenced to life, say, to death penalty, and they are injected with the cyanide, and the cyanide goes to block, this enzyme, cytochrome oxidase, then it creates such an anaerobic condition in the cells. So you can imagine that cyanide in large amounts in the body and creates a cellular, a suffocation of these cells, because then you're not going to have aerobic respiration, but rather anaerobic respiration, which will be producing just lactic acid. Obviously, over time, these cells will begin to die, and that means that tissues will begin to die. And once these tissues begin to die, organs begin to fail. And once organs begin to fail gradually, I mean, for instance, the heart, if it's not able to pump blood to all the parts, the cells of the various tissues dying off, then gradually it results into the death of the person. So that is how really active cyanide can be when exposed to it. It's such a chemical that's very potent that can cause fatality. Right. Another one is some microsystems. These also act directly on some enzymes, protein phosphatases. These protein phosphatases, you find them in the cell membrane, and you see, as you can see, these microsystems can cause some free radicals, formation of free radicals, and these then will result into the formation of some oxidative stress, some immune dysfunction, inhibition of some enzymes, and that, of course, results into some immunotoxicity. This has been found to cause such oxidative stress, immune dysfunction in some species of fishes, because these microsystems sometimes are found in the waste which is released into water bodies, and has been found to cause serious problems for some aquatic life. And that is due to a direct action that the microsystems will be having on these enzymes, some protein phosphatases. All right, now we can look specifically at indirect toxic action. I mean, looking at how this indirect toxic action can occur, different scenarios, what can happen in this case. So if we take a scenario 1, I mean, we have 4 different scenarios over here that we're looking at, hypothetical scenarios, where you have, first of all, you have this xenobiotic, which, I mean, is just being referred to as a chemical. And that xenobiotic is being converted by the cytochrome P450 into an unstable metabolite, and that unstable metabolite can actually bind to a cytochrome P450 enzyme, or let's just say an enzyme, still in the liver, inhibiting that enzyme. So and so there is a loss of enzyme activity. So a metabolite can become an inhibitor for an enzyme, and that will result into the impairment of a biochemical process. But this is occurring in the same cell, say, because I mean, this is hypothetical. This is a liver cell in which this is occurring. Now, another thing can occur where you have the xenobiotic, again, under the activity of a cytochrome P450 enzyme in the liver, gets converted into a metabolite. Now, that metabolite then crosses the nuclear membrane into the nucleus and binds to the DNA. So that is forming some DNA adduct that can result into some mutations in the genetic material. And that, of course, is going to affect gene expression, I mean, the synthesis of proteins there. So you see the difference between 1 and 2 is that in 1, the metabolite bound to a protein, because that's an enzyme. But in scenario 2, the metabolite bound to DNA. So that's at a genetic level. Now, if you take a look at scenario 3, what is happening is you are looking at the metabolism of xenobiotic in a particular cell. So let's say in one tissue of the liver, where you have the metabolism of the xenobiotic, and that the metabolite which is formed crosses the cell membrane into another cell, and it is in that cell that it goes to have its toxic effect. It even crosses the nuclear membrane of that cell, binds to DNA in a nucleus of another cell, second cell. So a cell of a different type is rather affected than the first cell, the cell type 1, where the metabolism of the xenobiotic actually occurred. And then scenario number 4, here you have metabolism of the xenobiotic occurring again under the activity of cytochrome P450 in a particular tissue. Now, after you have the conversion of such substance, you have a metabolite. The metabolite flows in the bloodstream to a different tissue. Here too, if you compare scenario 3 and 4, you realize that, I mean, the second cell which gets affected is actually just adjacent, and so you have the metabolite just crossing the cell membrane into that second type of cell where the effect is happening. But in scenario 4, you have the location actually being far apart, such that the metabolite flows in the bloodstream to a different tissue, where then it goes to have an effect on a different type of cell in a different type of tissue. Here too, it gets into the nucleus, binds to the nuclear membrane. Not necessarily only that, it may just be binding to some protein. So at the level of protein, you can have such a toxic effect occurring as well. Now we can go ahead and look at some examples of indirect action. Again, some more. I mean, today we're just looking at more and more of specific examples, so that it's not just a generalization of these things. So this is mechanism of action of di2-ethyl phthalate, which is a compound used as a plasticizer in the plastic industry. It's a common compound which is used, and it's known to have some toxic effects, particularly on the immune system, where you have DEHP, in the intestine can break down, undergo some hydrolytic reactions, and you do have this metabolite then inhibiting the process of differentiation, or let's say production of immune cells. So that, I mean, in the bone marrow, where you have the production of some of these immune cells that get developed, get differentiated proliferation, you have the inhibition of these processes. So, for instance, you have some of these immune cells which are being produced in the spleen. You have their numbers, being reduced due to the toxic effect of DEHP. And so you see that you have, for instance, interleukins, some interleukins, their production is inhibited, reduced, whilst the production of naive cells. So these are young cells. We do have what we refer to as, the immune system also has some memory cells that keep memory of some, let's say, previous infections and get adapted to those to fight them. And so if you do have in larger amounts naive cells than cells that have more of immune memory, then of course the immune system becomes weaker, and so you have such occurring, which then results into some allergic lung function as well, or a general impairment of the immune system. Right, so that's another mechanism based on that. We did look at acrylamide. I don't know, was it toxicokinetics or even before toxicokinetics? We saw an example of this, acrylamide, which you said it was endogenous. You can have acrylamide being produced endogenously, except that it's in small amounts. We also do have it in larger amounts coming from outside the body. And I did mention that when acrylamide is produced, particularly when you are preparing some fried foods in not so well ventilated kitchen, you can have the production of these things. Or even if you are frying, maybe put some potato chips or toasting bread, and it's bent, in this bent, so you can see on your screen that there's potato which is fried, which is normal, and there's some you see is sort of bent, and bread too. The more bent the bread is, the more acrylamide is in it. So you do have some toast. If it's over toasted, obviously there's some acrylamide in it. Not maybe not so much to have some toxic effects so much, but I mean, you can imagine if you have an overexposure to this over time, definitely it can result into some toxic effects. Now you do see two pathways of metabolism of acrylamide. You have the pathway up is epoxidation under the activity of the cytochrome P2E1. You remember we did refer to the cytochrome P2E1 to be, the most relevant cytochrome P450 enzyme, which is responsible for various toxic effects. If you go back to that lecture, you realize we refer to this particular enzyme as the most relevant in toxicological studies. And you see the formation of peroxide glycidiamidal, and from it, you have, some DNA adducts. That means it binds to some nucleic acid. It can bind to hemoglobin, meaning it will form some hemoglobin adducts there. Can also undergo some conjugation reaction with glutathione and produce some other toxic toxic metabolites, N-acetyl hydroxy carbamatocysteine. I mean, the names you see there, the keys are below, and so you can easily relate to them. Or its isomer is also produced, some N-acetyl S-carbamyl hydroxycysteine. All these can have some various toxic effects. Or the second metabolic pathway, where directly. So you see, you can quickly see that in toxicokinetics, where we did look at the phase 1 and phase 2 reactions, we did mention the fact that you have toxic substances that undergo, or let's say xenobiotics, undergo first phase 1 reaction before undergoing phase 2 reaction. And in some cases, you have xenobiotic undergoing only phase 2 reaction without any phase 1 reaction directly. So this is where you can have some of such example where you can see that first of all there is epoxidation. That is a phase 1 reaction, and then you start having conjugation reactions. So you have phase 1 reaction before phase 2 reaction in that reaction pathway. If you come below the second metabolic pathway, you realize that acrylamide goes straight into conjugation reaction, which is phase 2 reaction. It can bind directly to hemoglobin to form hemoglobin adducts, or binds to another endogenous substance, glutathione, and forms the N-acetyl carbamolecular cysteine, and further binding of that to some sulfoxide there. All right, so then what it means is that, see, this is one of the processes that you can refer to as toxication or toxification. Because this is not detoxification, a metabolism of a xenobiotic that results rather into toxification and not detoxification. This is a classic example of such. Now we do have also these compounds we refer to as benzopyrenes. You can see clearly, I mean, the structures look similar. They all have these 5 benzene rings fused together, all of them. These are benzopyrenes. I mean, a lot of it is, is found in the cigarette smokes. So we have 3 major pathways here through which there can be toxic activity of benzopyrenes. And the first pathway here is what we're referring to as a radical cation pathway. I mean, we have the pathways labeled below with the colors that are up there. So that the first pathway, where you have the benzo alpha pyrene being converted to a radical cation, and that is under the activity of a cytochrome P450 peroxidase. And that cation can then bind to nucleic acid, DNA, from some adducts, and that then will cause some damage, like some impairment to expression of genes that can result into later some lung cancers and things like that. The second pathway is diol epoxide pathway, where you have a metabolite So you see the benzopyrene, first of all, under the activity of some cytochrome P450 enzymes, and then some macrosomal epoxide hydrolase, undergoes some oxidation reaction, and the metabolite, a dihydrodiol, undergoes further oxidation. You can see there is some epoxide which is formed from it, and that epoxide can also form some DNA adducts. And that is where you have the toxic effects occurring with that DNA adducts. And then a third pathway, which is the orthoquinone pathway. What you see is that you have from that the dihydrodiol can have further oxidation to have a catechol, which is an unstable metabolite that can undergo that quinone pathway. I mean, you have these hydroxy groups that get converted into these cyclic ketone groups. That's a quinone that is being produced there, and such quinones can form nucleic acid adducts, or even binding with the endogenous compound glutathione, forming some adducts with it as well, which will then result into some toxic effects in the body. You do have even the oxidation process of this catechol, also resulting into some oxidative DNA damage there, because you have some free radicals can be produced in that process there as well. So that is indirect. So as you can see, you have metabolites along the line getting converted and binding, forming adducts with DNA along the pathway. So that's at the genetic level that you are having such a toxic effect. Now another is bromobenzene, which is a compound you find, say, in the production of these engine oils. You have bromobenzene, which is found in some different amounts depending on the quality of these of the engine oils. You have different amounts of these of this compound, the bromobenzene. Now you can see bromobenzene also can undergo some oxidation reaction, forming some epoxide. But the most important place which we should be looking at is that this central pathway, where under the activity of cytochrome P450, you have bromobenzene going into the, getting converted to bromobenzene 3,4-oxide, and this bromobenzene 3,4-oxide can have some covalent binding to proteins. That is forming some protein adducts, which then result into necrosis, which is also some form of suffocation of cells that once these cells are not having enough oxygen, it results into cell death. And so you have that as indirect, because it's the metabolite which is causing such a toxic effect, and not a bromobenzene itself. Of course, all these other metabolites have at different levels, different forms of toxic effects. Now, we see another example, the hexachlorobutadiene is also a compound which is used, also in the rubber industry, carpet, industry. You have the hexachlorobutadiene, which is used in the industry, that has indirect toxic effect on the kidneys. And when you look at all these reaction pathways where you have the hexachlorobutadiene, first of all, and you can see that there goes some conjugation reaction with glutathione, But I mean, the pathway which should concern us much is the pathway to the right, where first of all you can see there is some conjugation with a glutamine residue. I mean, you can see the enzyme there is a glutamyl transpeptidase. And so you have, first of all, some conjugation with an amino acid. I mean, just about 0.1 to 0.3% of the compound undergoes this reaction pathway. And further, you see that metabolite undergoes some further reactions of oxidation, and then even you see there is removal of protons from that metabolite, and eventually you have one of these intermediate metabolites that can have some covalent bonding with some biomolecules like proteins, DNA, and then you have the toxic effect. Just like we saw with the others, these metabolites forming some adducts, and these adducts then inducing some toxic activity in the biological system. Right. Naphthylamine is another example. We did see naphthylamine as one of these compounds, which is also used in industry, and we see that under the activity of cytochrome P450, if you take the reaction to the right, you see first of all there is some oxidation or hydroxylation reaction, because you can see clearly that amine group is now oxidized, and so in place of one of the hydrogens you have a hydroxy group. Then under the activity of the enzyme glucuronosyl transferase, you now have the conjugation of glucuronic acid. So that's a process of glucuronidation, a conjugation reaction. So you see, you have a phase 1 reaction first, and then now you're having a phase 2 reaction. And then from there on, you do have reactions, or even you can see there is rather some hydrolytic reaction, because there is a break-off of the glucuronic acid from that nitrogen there. That's terminal amine group, and you have the metabolite also forming some adducts with DNA, which then results into some bladder cancer. I think we did look at this particular example. So you have this bladder carcinogenesis. But on this other pathway, where you have first of all some phase 2 reaction, N-acetyltransferase acting, where you have conjugation of an acetyl group onto the terminal amine group. This N-acetylated metabolite does not bind to DNA, and so it does not cause any carcinogenesis. So that is one compound which is undergoing different metabolic pathways. And along one metabolic pathway, it can result into carcinogenesis. And along the other, it does not result into any carcinogenesis. Right. Now, another mechanism of toxic effect that you come across in toxicodynamics is what we refer to as lipid peroxidation. Lipid peroxidation. Now, you see, the cell membrane we have studied over HND and the BTech. You have studied the structure of the cell membrane. That the cell membrane is having what you refer to as the fluid mosaic model having phosphobiolipid into which you have embedded some proteins. Now, if that's the case, it means the cell membrane is mostly composed of lipids. Now you have this toxic substance, tetrachloromethane, or carbon tetrachloride. In fact, historically, I think sometime in the 1920s, 30s, the tetrachloromethane was used as a fire extinguisher in automobiles. So you see, you can imagine that's a toxic substance that wasn't known to have such toxic effect that it was being used as a fire extinguisher. So just like we have fire extinguishers in vehicles now, those times the fire extinguishers that were there, were made of this tetrachloromethane. Now, tetrachloromethane can get converted by cytochrome P450 embedded in a cell membrane and form some radical metabolite. And this radical metabolite can then trigger lipid oxidation. I mean, the oxidation of the membranes of the lipid. And such lipid peroxidation can result into the formation of different metabolites, like for instance, some isoprostanes. And these isoprostanes can react with some result into some biomarkers. We refer to them as being useful because we can make use of them as biomarkers for some oxidative cell injury, because of course we know it is out of lipid peroxidation that you have the production of this isoprostane. So that if you have some particular products which are known to be produced from isoprostanes, which we know are produced from lipid oxidation, then we can know that there is some form of oxidative cell injury that has occurred. Similarly, with this, pentane, also coming from that lipid oxidation, can also be used as a biomarker. But that is on a useful side. Those metabolites are used, as biomarkers. Now, the lipid oxidation can also result into, the production of other compounds which are noxious, other compounds which are toxic, like the 4-hydroxy nonenal or malon dialdehyde. Now, these compounds are toxic. So you do have this resulting into some reactive products. I mean, these are reactive products which can bind to some protein, causing some protein damage. Let's say you have this malonaldehyde or 4-hydroxy nonenal, which are reactive products which bind to proteins to form some protein adducts there, and this can result into cell death and some other toxic effects. So that is lipid peroxidation. So you can see it's a cascade of reactions there. That's why we do refer to it as lipid peroxidation cascade. Now let's take a look at another. I think this particular example we did see with paracetamol. Paracetamol, we did look at the various pathways, metabolic pathways of paracetamol, and we referred to the N-acetyl paraquinone, which it was responsible for paracetamol toxicity, because then we referred to the fact that it is that particular metabolite that forms some adducts with proteins and results into the toxic effects of the paracetamol. And here you see it clearly, this paracetamol, and one of the toxic, the toxic substance, toxic metabolite which is produced from paracetamol is that N-acetyl paraquinone, and that N-acetyl paraquinone actually causes damage to mitochondrial enzymes, The same way you have, even though you have other compounds which are produced in the metabolism of paracetamol, say for instance this 3-hydroxy acetanil, acetanilide, which does not have any toxic effect. You do have this quinone, and these quinones which are being formed, the N-acetyl paraquinones which are formed in this process, not only damaging these maybe mitochondrial enzymes, but even enzymes of the endoplasmic reticulum. So you see that is being presented as some protein damage that is occurring there. So it is these quinones that have toxic effects, particularly forming this damage or forming some adduct with these particular proteins or enzymes in the mitochondria or in the endoplasmic reticulum. Now, I think we will take maybe two more particular mechanisms of toxic effects, and one of them is calcium dyshomeostasis. So, what is that about? You have here a xenobiotic. So we take this as a liver cell in which we have a xenobiotic that is being converted by cytochrome P450 enzyme. I mean, that's an oxidation reaction where you have a reactive metabolite. Now, that reactive metabolite can then have some attack on an endoplasmic reticulum protein, which is actually responsible for the transportation of calcium. So it's a calcium pump, or say a calcium channel. Similarly, you have calcium channels also in the cell membrane directly, and so you also have such a reactive metabolite also impairing the calcium channel in the cell membrane. Now, in all of these, what then happens is that there is either an increase. So, for instance, if it is in the mitochondria, sometimes you do have what is calcium overload, which results into the breakdown of the mitochondria. A lot of metabolic processes occur in the mitochondria. That is actually the powerhouse, the energy house of the cell. So that if you have a lot of metabolic processes disrupted by such what we refer to as the dyshomeostasis, that's an imbalance of calcium in the cell, because you do have calcium as one of the cofactors of enzymes. That is another thing. Then you have calcium acting for the creating this potential across the cell membrane, where you have movement of ions across the cell membrane. And so that is another toxic activity, or the process by which you have toxicity expressed by some xenobiotics, calcium dyshomeostasis. Then, of course, I think we did speak much about this oxidative stress. So this is particularly just referring to the fact that you have a xenobiotic, which is undergoing some metabolic process reductase. So you have a quinone that can get reduced to a semiquinone. That's a radical form. It's like a free radical. And you do also have in the process of, say, mitochondrial respiration, where you have such radical quinones which are being formed, or let's say some free radical metabolites, which will be resulting into the production of some superoxides from oxygen, and this further will result into some DNA oxidation, protein oxidation, some form of damage to this DNA and proteins. I mean, that's at genetic level and at a protein level or at the enzymatic level. You have these reactive oxygen species which are resulting into such oxidative stress. We can zoom in on these particular processes of DNA oxidation and protein oxidation. So, I mean, for DNA oxidation, what you notice, I mean, DNA is made up of what? Nucleic acids. It's it's made up of nucleotides. And if you do have oxidation of the DNA, I mean, particularly oxidation of the nitrogenous bases, because for the sugar, the phosphate is constant for all these nucleic acids, DNA. You have the nitrogenous bases undergoing some oxidation reaction, which will be resulting, and that oxidation reaction is actually occurring because of those free radicals that have been produced in the system. And so you have, say, from guanine, you have something like, an oxidized form of guanine as oxoguanine, or you do have, the ring even opened. That's a metabolite of the nitrogenous base. Obviously, these are not nitrogenous bases in the nucleic acid. So that's some kind of a damage to the DNA. The same way you have protein, consisting of amino acids. So for instance, you have these amino acid systems undergoing some oxidation reaction, again, under some, the presence of, say, hydrogen peroxide, an antioxidant, or say an oxidant, which is in a system, and that can result into a breakdown of cysteine, or you have the formation of the cysteine sulfonic acid. So there's some kind of oxidation occurring there, or cysteine disulfide, where you have two cysteine molecules conjugating together. All these will result into some deactivation of the proteins. And that is coming from, the previous slide where we were looking at the oxidative stress. We just zooming in specifically on that DNA oxidation and protein oxidation to see what really is happening. And you see in the DNA, it is actually the nitrogenous bases that are getting oxidized. And you have in the proteins, some amino acids, get oxidized or even get conjugated in the process. Now, we also have, some processes of cell suicide, so cellular suicide. I mean, the process of apoptosis is cellular suicide. That's how it's referred to. And you see here, I mean, if you look at the reaction pathways are numbered, so you can actually go around one all the way to the last. And you see, you have a xenobiotic, again, being converted by cytochrome P450 enzyme into a reactive metabolite. And that reactive metabolite can cause some DNA damage. And that DNA damage then, begins to, let's say, some kind of a mutation. And you have the expression of some protein factors that then bind to mitochondria, and in that process, those protein factors even further go to activate a cascade of reactions, producing other, apoptotic factors. I mean, those are proteins like the Cas9, which sort of what's being referred to as apoptosome, an assembly of what is referred to as a wheel of death. You have these protein factors that are forming a complex together, which out of them you then have the production of even other proteins, protein caspases, which go into the formation of, say, what is referred to as executioner capsaces. These are also some proteins or some factors that actually, carry out like internal cell digestion. I mean, sort of feeding on the cell. It's like the cell causes its own digestion, its own destruction. The cell serves as a substrate for these capsaces, and that then results into apoptosis, which is what you're referring to as suicide. I mean, apoptotic cell death. I mean, this usually happens. This is one of the processes that happens with some cancer cells. So, for instance, you have some apoptotic sort of a protection by cells, so that in some cases, when you have cells that are infected, I mean, they have some carcinogenic gene, and they wish to save other cells from themselves, so that because they know if they reproduce, then it will result into the death of the other cells. And then these cancer cells actually can carry out this process of apoptosis. And some medications are actually being developed based on this process of apoptosis, so that cancer cells will begin to cause their own death, give a certain protection to other cells from getting infected. All right. When we take the last one, which is a stress responsive kinase signaling, and here too we see we have a xenobiotic. Again, under cytochrome P450 enzymes undergo some oxidation and has a reactive metabolite produced, and this reactive metabolite then becomes an inhibitor to this phosphatase inhibition. I mean, the phosphatase inhibition, then results into some nuclear response where you have some immediate genes which are produced in response to such inhibition of these enzymes. And from the nucleus, you have the proteins which are expressed from this response, then triggering that process of apoptosis. You also do have production of these some of these proteins, some of these factors, resulting into increase in the permeability of mitochondria, so that you have the release of apoptotic compounds as well. And just like we saw with those apoptotic factors like Cas9, you have some other factors also being released from the mitochondria, which will also trigger or facilitate the process of apoptosis. So these are different mechanisms. Of course, not exhaustive. If you go further and further, you will find more and more of these mechanisms of action of toxic substances. If you go into research in toxicology, obviously you go deeper and deeper. You are studying a particular compound. You may come across a particular process of toxic effects which has not been discussed in literature, or maybe there's some similarity, but there could be something which is peculiar with a particular toxic effect of a substance. So this is just to whet your appetite on the various mechanisms of toxic effect of different toxicants, so that based on this, as you go ahead, go forward, you can decide which field, if you want to go into research in toxicology, and you want to actually study mechanisms of toxic effects like toxicodynamics, you come across some of these things. All right, so I thank you so much for your audience. If there are any questions, the chat is active. You can write in the chat. I'll respond to you. Alternatively, you can raise your hand. I will enable your microphone. Then you unmute yourself, you speak, I'll respond. The floor is open.